Good morning from California and good evening to all of you. It's my pleasure to be here today to talk about the evolution of antibody drug conjugates. So I loved the way that Dr. Bapsy wrapped up her presentation by talking about the importance of research, because this is actually what I've spent, as you just heard, the last 35 years doing. And so the last 20 years working on ADC. So it was a really interesting journey for me to put together this presentation about the evolution of antibody drug conjugates. And I need somebody to either advance the slides or tell me how to advance the slides. Okay, thank you. Let me put this in full screen. So I really wanted to start from the very beginning with really how targeted therapy began. And this is with around in 1900, the German scientist Paul Ehrlich, who discovered many things. Sorry, I can't see the whole slide. There, oh, thank you. it was cut off, thank you. So Paul Ehrlich really had the first idea of targeted therapy that he called the magic bullet hypothesis. And we use this term a lot now, but it actually originated more than 100 years ago. He postulated the existence of cell surface receptors. He postulated the magic bullet concept of drugs going to an intended target. He even postulated that antibodies could be magic bullets that identify a target without harming the organism. And he even back then envisioned attaching toxins to antibodies to improve therapeutic activity. Next slide, please. And I know this is a little bit difficult to see, but this is a timeline of the findings of Paul Ehrlich. And I think what I wanted to just highlight was in red, I don't know if I have a cursor or not, I guess I don't, that in the bottom red was a discovery of, okay, hang on. No, I don't have a cursor, okay. Okay, so there was the discovery of oncogenes, which is circled in the top on the red. And then in the bottom, the large box is a very important discovery by Kohler and Milstein, which is the discovery of how to make monoclonal antibodies. And then shortly thereafter, if you look at the two red boxes, you can see very importantly the discovery of both the EGF receptor and HER2, and documentation that these molecules were actually oncogenic. And then following that were the development of antibodies to both EGF receptor and HER2, the approval of the first antibody for therapy, which was rituximab, and in 1998, the approval of trastuzumab, which was the first approved antibody for a solid tumor indication, which was HER2 positive breast cancer. Next slide, please. So here's a timeline of approved ADCs. And you can see that this has been going on for a long time. So in fact, in the year 2000, the first approval happened, which was with Mylotarg, which targets the CD33 antigen on AML cells with a potent DNA damaging agent. And I'll go into the different antibodies and cytotoxic drugs later. There was a gap of about 10 years, and then very interestingly, Mylotarg was actually withdrawn from the market 10 years later in 2010. And this was because the post-marketing studies that were mandated by the FDA did not support the preliminary findings of better efficacy versus safety. And in fact, there were a lot of safety signals that warranted withdrawal from the market. Just a year later, another ADC was approved, which is Adcetris for Hodgkin's disease. And again, I'll talk about the targets and the drugs in a bit. A couple years later was Kadcyla, approved for HER2 positive metastatic breast cancer. And then there was a bit of a gap between 2013 and 2017. There was a lot of activity during these years, a lot of preclinical and clinical investigation, but it was a few years later before the next ADC approval. And then starting in 2017 and moving forward, there have actually been a flurry of ADC approvals, which is really gratifying for those of us that have been in the field for so long. So very interestingly, Mylotarg was reapproved in 2017 with a more restricted label. Besponsa was approved for ALL. In 2019, Kadcyla got the indication for treating adjuvant HER2 positive disease in patients that have residual disease. Polivy was approved for diffuse large B-cell lymphoma, and HER2 was approved for HER2 positive metastatic breast cancer. And then in 2020, we have Blenrep, the most recent one, for multiple myeloma, Padcev for metastatic bladder cancer, and Trodelvy for metastatic triple-negative breast cancer. So just in 2019 and 2020, we've actually had almost more approvals than we have had for the last 20 years. So the evolution of ADCs really shows us that it's taken a long time to get here, but now we are actually starting to see a good number of successes. Next slide. So I think you all know this, but very simplistically, this is what an antibody drug conjugate is composed of. It's composed of an antibody with a linker that links it to a cytotoxic drug. Next slide, please. And the mechanisms I think you're all very familiar with are that you get receptor binding of the ADC to its target. This can also be a mechanism, as I've noted, of antibody-dependent cellular cytotoxicity. But I think more importantly, the binding of the ADC to the receptor elicits receptor-mediated internalization into the cell. This complex is trafficked to the lysosomes, where the ADC is catabolized to release its active free drug. Now, this bottom cartoon shows that if the free drug is a microtubule binding drug, it will go to the microtubules to elicit its mechanism of action. If it's a DNA active agent, then it has to cross the nuclear membrane to get into the nucleus to elicit its mechanism of action by binding DNA or some of the DNA enzymes. And then importantly to note that I've shown here as well, that if an ADC has a cleavable linker that releases a cell permeable cytotoxic drug, the cell permeable drug can exit the cell to kill neighboring cells, which is called bystander activity. And I'll actually get back to this in a minute. Next slide, please. So going through the different components of the antibody, of the ADC. So first you have the antibody. The most important criteria of the antibody is that you have a target that is tumor selective. In other words, there's high expression, in most cases, high expression in tumor versus normal. There are different forms of antibodies, which I've listed here. What I've done in this slide is I've bolded and put in italics the different forms of ADCs that are in approved ADCs. So there are ADCs that are approved that have both the IgG1 and the IgG4 format. And then with the linker, there are many different types of linkers, and they are linked to the antibody in different ways. So this shows at the top, they can be linked through cysteine, or they can be linked through the amine on lysine. There are cleavable linkers that are either peptide linkers cleaved by proteases, or there are acid labile linkers cleaved by the acidic pH in lysosomes. Peptide linkers and acid labile linkers are present in approved ADCs. And then there are different non-cleavable linkers as well. And the example here is a thioether linker that's found in TDM1. Many different types of cytotoxic drugs have been assessed. There are different tubulin binders, maytansinoids and auristatins, again, are present on approved ADCs. There have also been tubulizins that have been studied as tubulin binding drugs. DNA binders have sort of come into focus more lately. Calicheamycin was the first one, and it is the drug on Mylotarg, but there have also been other classes of DNA binding agents that are called PBDs, duorchomycins, and then the topoisomerase 1 inhibitors, particularly camptothecins, which are present on 2 of the recently approved ADCs. So there's many things to choose from, and the bottom line is you need to get all 3 right. And there's no logical way to test these. You have to actually go through an iterative process of just testing antibodies, linkers, and drugs, and looking at efficacy and safety until you find that sort of magic combination. Next slide, please. So, and click again, please. So the first generation of cytotoxic agents, as I mentioned, were the calicheamycins. These induce DNA strand breaks. Again, this is the drug that's on the first approved ADC, Mylotarg. These were developed originally by Wyeth, and who was then acquired by Pfizer. So Pfizer really has been the company that has focused on calicheamycins. Another type of cytotoxic agent are the auristatins, and the auristatins were pioneered by a company, Seattle Genetics. They have investigated auristatins with many different partners for both preclinical and clinical development. And there are 3 approved drugs that have auristatins. One is Polivy, one is Padcev, and one is Blenrep. The auristatins are comprised of either mmae or an analog of this, mmae, which is cell impermeable, which is actually on Blenrep. And And then there are the maytansinoids that were pioneered by the company ImmunoGen. And again, ImmunoGen, like Seattle Genetics, had many, many different partners for both preclinical and clinical studies. And the one approved ADC with a maytansinoid, which is DM1, is Kadcyla. Next slide, please. So let's look at how these drugs have been linked with different linkers to the antibodies. So the first generation of ADCs had different types of cleavable linkers. So if you look at the top, this is the structure of both gemtuzumab ozogamicin, which is Mylotarg, and inotuzumab ozogamicin, which is Besponsa. So only the antibody here, the targeting antibody, is different. One for CD33 and one for CD22. The linker and drug are the same. The drug is calicheamycin. The linker is this, what we call ACE-bute linker. It's actually cleaved at the hydrazone, which is where you see that double bond near the nitrogen to release the free drug. So this is an acid labile linker. The other type of cleavable linkers is shown on the bottom, which is the valine citrulline linker. So this is a dipeptide that's cleaved by proteases that are selectively expressed in tumor cells to release the free drug mmae, which is, again, a tubulin binder. There are 3 approved ADCs that have this same linker drug, which is Adcetris, Polivy, and Padcev. For 3 different targets, the same linker drug, all with approximately the same DAR. And I will mention DAR is drug to antibody ratio, so how many drugs there are to antibody. Most of these have a similar DAR. Next slide, please. And then there are what we call first generation ADCs with uncleavable linkers. So very recently, this ADC was approved, Blenrep. This is for multiple myeloma. It targets an antigen called BCMA. And this has an uncleavable linker shown as MC. And this has the auristatin MMAF, which again is a auristatin that is not cell permeable. So once this ADC is catabolized inside the cell, the MMAF cannot exit the cell. So this would not have bystander activity. And these types of ADCs have very different properties, which I'll cover at the end. The other ADC with an uncleavable linker I think everybody is familiar with is trastuzumab emtansine, or Kadcyla. It targets HER2. It has the MCC uncleavable linker and the drug DM1. Next slide, please. So I wanted to talk a little bit about the difference between uncleavable and cleavable linkers. This is really important to think about in terms of the mechanism of action of ADCs. So here I show on the left, if you have an ADC like TDM1 or like Blenrep, for instance, which I just mentioned, that have uncleavable linkers, these antibody drug conjugates go into the cell. They go to the lysosome where they undergo lysosomal degradation to actually release a catabolite that is not the free drug. The catabolite is actually the lysine from the antibody, so in this case from trastuzumab, if it's TDM1, the lysine to which the linker drug is conjugated. So they have lysine, linker, drug. And in this case, lysine, MCC, DM1. If it was Blenrep, it would be cysteine, MC, MMAE. These catabolites in the lysosome actually have to be transported out of the lysosome by very specific transporters, which I've shown it with the little squiggly lines on the cell, on the membrane of the lysosome. And so once this catabolite is released, and here it shows lysine, MCC, DM1, this cannot exit the cell. So these uncleavable linkers generate catabolites which are not cell permeable. In contrast, if you look on the right with ADCs with cleavable linkers, this can be disulfide linkers that are undergo reduction to release the drug, peptide linkers that are released with proteases, and the acid labile linkers, which I discussed. These actually are catabolized to give your amino acid linker drug in the lysosome, and then the linker, which is shown here with the R's, the linker is cleaved to give the free drug. One more click, please. Which then will exit the cell and can cause cell death of neighboring tumor cells, and this is called the bystander effect. So it's very important to keep in mind the different sort of metabolic fates of ADCs with uncleavable versus cleavable linkers. Next slide, please. So I'm going to spend just several slides giving you sort of snapshots of the development of different ADCs. And again, this will, I think, emphasize how different each approach has been. So first of all, we can talk about the development of TDM1, which was the first HER2-directed ADC. So the antibody that was selected was trastuzumab. For the cytotoxic agent, so this work was done at Genentech, the cytotoxic agent, actually maytansinoids and auristatins were heavily studied. And after much study, it was concluded that the ADCs with DM1 were better tolerated versus the auristatins, and I'll talk about this in a minute. And then we also looked at cleavable versus uncleavable linkers. So again, you kind of have to do this iterative approach to figure out what combination of these works the best. So this shows where we started. So we partnered with ImmunoGen because they had the technology for maytansinoid ADCs. And the flavor of the day for these ADCs were ADCs with disulfide linkers, which are shown on the left here by SPP. So that designates the disulfide linker. So you can see the disulfide, and to the right of it, you can see a black line going down, which indicates a methyl group. This was the type of linker that was studied historically for all DM1 containing ADCs. And the MCC, uncleavable linker, for different targets was actually shown not to be active. And so we actually at Genentech took trastuzumab SPP DM1 into preclinical safety studies and found that we got unacceptable toxicity with this disulfide linker. So we had to sort of go back to the drawing board. Next slide, please. Okay, go one more, please. And so what we did was we liked the antibody trastuzumab and we liked the drug DM1. What we did was a series of investigations into the linker. And so what this shows, going from top to bottom, are the different linkers that were investigated on trastuzumab with a maytansinoid drug. And you can see, and I've designated with the red line, going from top, there is no methyl substitution around the disulfide bond, to SPP where there is one, to the next one where there are 2 methyls, to the next one where there are 3, and finally the MCC linker, which is uncleavable. And the addition of these methyl groups on either side of the disulfide bond cause enhanced stability, enhanced linker stability, which is indicated by the arrow on the right. So the more kind of steric hindrance you have around the disulfide bond, the more stable the linker is. And of course, the MCC is the uncleavable linker, which is the most stable. So what happens with all these linkers? Next slide, please. So if you look on the left, we looked at these in a HER2-positive breast cancer cell line, and there was no difference. All of the linkers on trastuzumab DM1 behaved all the same, which you can see. Oh, only 10 minutes. Okay, I better speed up here. You can see on the left-hand side with the cell line. And suffice to say that the stability of the linker conferred increased stability in a PK study in the middle and increased efficacy in mouse tumor models on the right. Let's click through the next slide if I only have 10 minutes left. So go one more. These are just preclinical data, so we can skip through these. So ultimately, one more, please. The outcome of all these studies was that the uncleavable MCC linker gave us great efficacy and better safety, and as you know, was approved in second-line treatment as well as the adjuvant setting. So I wanted to spend a minute talking about ADCs for non-Hodgkin's lymphoma. So if you think about B-cell malignancies, there are a lot of different targets to choose from. So these are shown here. You can see CD19, 20, 21, 22, 72, 79, et cetera. Next slide, please. And so, again, this was an iterative process. On the left here, it shows CD79B versus CD22 with either valsat mmae or MC-MMAF, and the top left shows that the valsat mmae works better. Several different targets are shown on the right, 4 different B-cell targets, these with the DM1 containing, and in three of 4 of these, the SPP DM1 was more active than the MCC DM1. So all of these different combinations were investigated on different B-cell targets. Next slide. And let's just go to the next slide. We don't have to go over this. So bottom line is that the valsat mmae was chosen over the DM1 containing ADCs. And interestingly enough, 2 different NHL ADCs went into the clinic. Both had valsat mmae, one targeted CD22, one targeted CD79B. And what is shown here is that if you look in red, the CD79B ADC outperformed the CD22 ADC, so that was moved forward. Next slide. And the results of the registrational trial are shown here. This ADC is called polatuzumab vedotin. It was in a trial where it was combined with bendamustine rituximab versus BR alone. And you can see clearly from these Kaplan-Meier plots with the blue line that polatuzumab in this combination gave much better PFS and OS compared to BR alone. And this was the study that the approval was based on. Next slide. And so just very quickly, for second generation ADCs, next slide, please. And just click 2 more times so I can do this all at once. So if you look on the left, this is how Kadcyla is made. It is made by linking in a random way the linker drug to lysine residues. And as we all know, you get a heterogeneous distribution of drug. All of the mmae, the auristatin containing ADCs are linked through reduction of the antibody interchain disulfide bonds. So they're all in that hinge region, which is shown here. So you get approximately a DAR of 4, but you still get a heterogeneous distribution. And what antibody engineering has allowed us to do in recent years is to actually engineer in to antibodies specific cysteine residues into an antibody so that you can get precisely, this shows a DAR of 2, or if you engineer in two sites for conjugation, you can get precisely a DAR of 4. So this has really changed the way that we think about conjugating so that we can get away from this heterogeneous mix of DAR. Next slide. And can you click a couple of times? So I'm just going to show this really quickly. So there's been a ton of work done over the last probably 8 to 10 years on a new class of DNA damaging agents called pyrrolobenzodiazepine dimers. They alkylate and cross-link DNA. There's 2 that are shown here that just have different linkers. One's called talarine and tesserine. 1 click, please. These are more potent than tubulin binding agents. They're active on dividing cells and non-dividing cells, which is different from tubulin binders. And they've all been tested with cleavable linkers, so they have bystander activity. Next slide. And these PBD dimers have been tested on a number of different targets. The targets are shown here, both hematologic and RTKs, as well as other. These are super potent payloads. They have a lot of different toxicities, which are listed here. And if you look in the box here, you can see that the MTD is actually in the microgram per kilogram amount. So this indicates how unbelievably potent they are. And the bottom line here is that most of the ADCs with PBD dimers that have gone into the clinic have been terminated because they get toxicity without achieving any kind of efficacy. There are still some being studied, so stay tuned, but no successes thus far. Go on to the next slide. Let's go ahead and, I don't have much time. I think I'll have to skip through this. Let's go one more and go one more and go one more. And then I think I just want to show, no, go back one, please. I just want to show this slide to depict the challenges of developing ADCs. So this is a timeline of ADC development. And in the purple bars, it shows new ADCs. And in the gray bar, it shows discontinued ADCs. And then in the green bar, it shows approved. And so you can see the number of new ADCs has really increased over the years. And this is through part of 2020. But you can also see that the number of discontinued ADCs as we get into, especially 2018 and 2019, is almost as much as the new ADCs that have been undergone development. This is a very, very challenging type of drug to develop. And I would safely say that half of the ones that go into the clinic have to be discontinued, mostly for safety issues, some for business strategy issues. Next slide. And 2 more, there we go. And so I just want to just show this very quickly, that we think that the primary problem with this is that the ADC processing leads to unwanted toxicity signals. So ideally you get binding to your tumor cell and you get tumor cell killing. But if you have a labile linker, which is shown on the left, you get free drug in the circulation. And on the right depicts uptake into normal cells. This happens with every ADC. There are lots of normal cells that will non-specifically take up antibodies and ADCs. And so you can get target independent toxicities, which is very, very difficult to control. Next slide. Okay, just two more examples of the newer ADCs that I think people are really excited about. One, of course, is trastuzumab deruxtecan. This is an ADC that has a 4 amino acid linker in contrast to the 2 amino acid linkers that are on valsat mmae. It's got a topoisomerase 1 inhibitor. These topo 1 inhibitors are much, much less potent than the tubulin binders or calicheamycin. And so therefore they can actually load up the drugs to get a DAR of 8. This molecule has been shown to be active in preclinical models in both HER2 positive and HER2 low. Next slide, please. And very impressive clinical data, again, in both HER2 positive and HER2 low metastatic breast cancer. So the preclinical data very nicely translated to the clinical data. The last ADC I'll talk about is the trastuzumab govitecan. This is the one that's approved for metastatic triple-negative breast. This has a super complicated linker, which is shown in the middle here. But ultimately, in the green part, it shows a pH-sensitive part that is actually cleaved. This is a very unstable linker. It releases a lot of free drugs, so therefore the ADC has a short half-life. And the toxicities of this ADC are exactly what one would expect from an irinotecan type drug. And again, this is an irinotecan derivative as the drug. These are low potency drugs, so they also have a DAR between 7 and 8, and again have shown very impressive clinical activity in metastatic triple-negative breast cancer, which is where it was recently approved. Okay, conclusion slides. So just overall, I just want to say the development of ADCs is really challenging. There's toxicity due to non-target mediated uptake. This, I think, in my opinion, remains the biggest challenge and limitation. For calicheamycin ADCs, there are very serious safety signals, and dosing is limited to the very low mg per meter squared amount. With mmae containing ADCs, we get similar safety signals regardless of the target. So these are not target dependent safety signals. These are due to the payload. And as you would expect with a tubulin binder, you get neutropenia and peripheral neuropathy. And the dosing actually is limited to less than 2 mg per kg. The new mmae containing ADC, which is a non-cleavable auristatin, actually very interesting, has a different safety profile than mmae. Again, it generates this cell impermeable catabolite. And in this case, the safety signals are ocular toxicities and thrombocytopenia. And then, of course, with TDM1, the DLT in the clinic was thrombocytopenia, which limited dosing to 3.6 mg per kg. We were hoping to go higher. That wasn't the case. And just to reiterate, the highly potent cytotoxics, such as the PBD dimers, have really not been developable to date in terms of there are no approved ADCs with a PBD dimer. And the final slide. Oh, one more click, please. So these newly approved ADCs that I just talked about, I think show very promising approaches. So these reduced potency topo-1 inhibitors, again, they have sub-micromolar potency compared to picomolar or nanomolar potency in vitro assays. This allows for a high DAR, so you can get 7 to 8 drugs per antibody. And I think this is reflected in the higher clinical doses. With trastuzumab deruxtecan, it's 5.4 mg per kg. And with trastuzumab govitecan, it's 10 mg per kg on day 1 and day 8 of a 3-week cycle. I think the other really cool thing about these new ADCs is that these targets are expressed across a wide variety of solid tumor types. And so trastuzumab govitecan was approved in triple-negative breast cancer, but there are ongoing trials in ER positive, in bladder cancer, endometrial cancer, lung cancer, head and neck cancer, and there are combinations with checkpoint inhibitors and PARP inhibitors. The same with enfortumab vedotin. This is the one that targets Nectin-4, and it was approved in metastatic bladder cancer. It's also being tested across multiple solid tumor types because the target is expressed across multiple solid tumor types. And again, is also undergoing combination studies with checkpoint inhibitors and chemo. And then trastuzumab deruxtecan also is looking at HER2-low, gastric cancer, lung cancer, and colorectal cancer, and is investigating a variety of combinations with both checkpoint inhibitors, targeted agents, and chemotherapy agents. So that concludes my talk. Again, it was my great pleasure and privilege